Very good afternoon everyone. In our previous classes we have discussed about different kinds of tissue system and we have seen different kinds of tissues. So now in this class we are going to discuss about how xylem and phloem are arranged in a vascular bundle. So before going into the topic, I want to recollect all the things that we have learned in this chapter. So it will be like a revision session. So there are two main kinds of tissues, the meristematic tissue and the permanent tissue. The main difference between them is here they have the ability to differentiate into different kinds of tissue. Here they don't have the ability to divide into different kinds of tissues. So they are permanent. So if we take a parenchyma, it will divide into another parenchyma. That's it. But here, if we take an apical meristem, it has the ability to differentiate into different kinds of other tissues. So that's the basic difference between them. Here they have the ability to differentiate and here they don't have the ability to differentiate. So that's the basic thing. And coming to tissues, there are many tissues. So it will be confusing for us to learn and remember. So let's divide them into groups. So let's suppose that the first group belongs to meristematic tissue and the second one belongs to simple permanent tissue and the third group we will name it as complex permanent tissue. So it will be easy for us to remember and recollect all those things during exams. So let's start with the first group, meristematic tissue. So here there are three different kinds of meristems: apical meristem, intercalary meristem and lateral meristem. Apical meristem, these are present at the tip of the root and stem. Remember, tip at the tip of the root and stem. That's the main thing for them. And coming to second one, intercalary meristem. These are present between two mature tissues. These are present between two mature tissues. So if this is a mature tissue and this is a mature tissue, it is present between them. Coming to third one, lateral meristem. It has many kinds of tissues in them. Intravascular cambium, vascular cambium, core cambium and intervascular cambium. So except intravascular cambium, all of them are related to secondary growth. And here apical meristem and intercalary meristem are related to primary growth. So here I have mentioned two terms, primary growth and the secondary growth. The growth which happens during the initial phase of life is known as primary growth. That primary growth we can see with the help of apical meristem and intercalary meristem, and in lateral meristem only with the intravascular cambium. Right there. So, primary growth can be only seen in apical meristem, intercalary meristem and intravascular cambium meristem. And the rest of the things like intervascular cambium and vascular cambium and core cambium, they are responsible for secondary growth. Right. So we have finished the first one. The second group, simple permanent tissue. And we can also say them as homogeneous. Why? Because here they have only one kind of cells. If we take this complex permanent tissue, they are heterogeneous. The main difference between them is here they have only one kind of cells. They have, if we take parenchyma, they have the only parenchymatic cells. But if we take xylem, they have different kinds of cells. That's the reason why they are known as heterogeneous and these are known as homogeneous. So remember that thing. And coming to the second part, simple permanent tissues. Here we have parenchyma, collenchyma and sclerenchyma. Parenchyma, these are primitive and fundamental and the most common tissues that we will see. And whenever you find in an MCQ or somewhere else, if you find that they have intercellular spaces or thin-walled cells, then close your eyes and tick the option parenchyma. That's the main things for them. So there are many other things we have discussed them in our previous class. And coming to collenchyma, let's compare them. If we compare them, we can understand and we can remember for a long time. So collenchyma and sclerenchyma, both of them are for mechanical support. But the main difference between them is here collenchyma are living cells, but the sclerenchyma are dead cells, right? And here the lignin element called lignin is absent, and here the lignin is present. So the collenchyma are living cells and the sclerenchyma are dead cells, and here they don't have lignin, and here lignin is present in the walls. So that's the basic things about them. And coming to the third part, permanent complex permanent tissue. And as I have told you earlier, these are heterogeneous in nature, because if we take xylem, there are 4 different kinds of elements in it. If we take phloem, it has another 4 kinds of elements in it. Xylem, except this, all these 3 elements are dead elements, and the only xylem parenchyma is a living element. So the tracheids, vessels and xylem fibers are dead elements, and the xylem parenchyma is a living element. The main function of xylem parenchyma is radial conduction. Radial conduction of water. So the xylem parenchyma, the main function of it is to conduct the radial conduction of water. Coming to tracheids, these are narrow in nature. They have narrow lumen, and they have, coming to vessels, xylem vessels, they have wider lumen. That's the basic difference between them. They have narrow lumen and these have a wider lumen. And coming to xylem fibers, they support both the tracheids and vessels. They give the support to them. That's it. That's their function. This is present in the secondary group. So tracheids and vessels are known as water conducting elements. So the main function of xylem is to conduct the water with the help of tracheids and vessels. The support is given by xylem fibers. And there is one more thing, xylem parenchyma, which is a living thing, just an exception. All the 3 are dead, and the xylem parenchyma is a living thing, living cell, and it helps in radial conduction of water. And coming to phloem, here we have sieve cells. Sieve cells are present in pteridophytes and gymnosperms, and sieve tube is present in angiosperms, and same with companion cells. This is only present in angiosperms. So just like the function of tracheids and vessels, sieve cells or sieve tubes does the same function. The difference is the xylem conducts the water from roots to all parts of the plant, and the phloem conducts the food from the leaves to all parts of plant. So this is done by sieve cells and sieve tubes. So with the help of companion cells, the both are like sister cells. The both are almost attached to each other with the help of plasmodesmata. And this phloem fibers help these sieve elements. Here xylem fibers help the tracheids and vessels, and here phloem fibers help the sieve tubes and sieve elements and companion cells. And coming to phloem parenchyma, they help in radial conduction of food. Here if we see xylem parenchyma, here it helps in radial conduction of water. But here the phloem parenchyma, almost the same function. Here it is replaced by food. So that's the basic difference between them. So let's go to the other part. Now we will be talking about the arrangement of xylem and phloem. Enlarged condition. What happens here is the first formed xylem, which is also known as protoxylem, inside and the metaxylem is formed outside. And in Exarch condition, also known as centripetal condition, the metaxylem is present inside and the newly formed protoxylem is present outside. So in Exarch condition, also known as, in Endarch condition, also known as centrifugal. Here the protoxylem is present inside and the metaxylem is present outer side of the protoxylem. And in Exarch condition, also known as centripetal condition, the metaxylem is present inside and the protoxylem is present outside. And we have radial vascular bundles. So xylem and phloem together are known as vascular bundles. In radial vascular bundles, here the xylem and phloem are arranged in different radii. Of xylem and phloem. So they are not present in the same radius. They are present in an alternative way. So we will see this kind of thing in roots of dicot and more. So if they have the radial vascular bundle condition, they will have the Exarch condition. The next condition is known as conjoint collateral vascular bundles. So this is exactly opposite to this. Here the xylem and phloem are present in the same radius. Xylem and phloem on same radius. And the conjoint collateral, if they have this condition, they will have the Endarch condition. So the main difference between them is, in radial vascular bundles, the xylem and phloem are arranged in different radius. But here, in the conjoint collateral vascular bundles, the xylem and phloem are present on the same radius. This we can see in Endarch conditions, and this we can see in Exarch conditions. And here again, it is of two types, open and closed. In open, if we take this as a vascular bundle, this is phloem and this is xylem. In between, they have a structure called cambium. Which is helpful in secondary growth. But here they don't can be seen in monopods. And here this can be seen in dicots. Dicot stem, dicot root, dicot stem. Sorry, not root, dicot stem. So this is the reason why the dicot stem will have the secondary growth, and the dicot stem doesn't have secondary growth because they don't have cambium. So we will understand them when we talk about monopod stem, monopod root, and dicot stem and dicot root. So we will understand what these things are. So in attached condition, the protoxylem is present inside, which is covered by metaxylem. But in exarch condition, the metaxylem is inside and it is covered by protoxylem. And coming to radial vascular bundles, here the xylem and the phloem are present on different radius. When this condition is present, exarch condition is also present. And in conjoined collateral vascular bundles, the xylem and phloem are arranged in the same radius. When this condition is present, exarch condition is also present. And again, conjoined collateral vascular bundles are divided into two types: open vascular bundles, open conjoined collateral vascular bundles, and closed conjoined collateral vascular bundles. In open, they have the cambium. That's the reason why the dicot stem will see the secondary growth. In closed, they don't have any cambium. That's the reason why the monopods doesn't see any secondary growth. And again, we have some other things like bicollateral thing. Here, the phloem, this is xylem. On the both sides, it is covered by phloem. And also on the both sides, they have a strip called cambium strip. And this can be seen in Solanaceae family. So this is known as bicollateral conjoint. Bicollateral. And we have one more. Amphicribral and amphivesical. If the xylem is present inside and the phloem is present outside, then it is known as amphicribral. Xylem and phloem. So the xylem is present inside, which is covered by phloem. Then it is known as amphicribral. If the xylem is present outside and the phloem is present inside, then it is known as amphivesical. So that's it for today. Thank you for listening to me. See you in the next class.